Good morning and welcome to the kickoff of our 2014 lecture series. I'm Carrie Lewis, Executive Director of DCA, and I'm delighted to see all of you in the house this morning. Before we begin, we'd like to thank our sponsors, Dahl Law Firm and Ross Estate Planning. Please give these folks a round of applause. We're also thankful for the support of our lodging partner, Apple Creek Resort, and our coffee host, Base Camp Coffee. This morning, we are pleased to welcome Dr. David Hunnicutt to Door County. Dr. Hunnicutt is an associate professor of biology at St. Norbert. He did his undergrad work at Augustana, his master's at Illinois State, and he received his PhD from the University of Wisconsin-Milwaukee. And since I am currently playing host to some sort of head cold or something, I'm going to keep it short and just turn it right over to Dr. David Hunnicutt.
Good morning. Can everyone hear me? No? Yes? Now you can't? Okay. I'll be honest, I'm not used to working with a mic. As a microbiologist, I don't have a lot of call to work with the microphone. We're a small and deeply disturbed group, I guess. I wanted to thank the Door County Auditorium for inviting me up. I've never been up for one of these, although I have colleagues who have and have spoken very highly of it. I want to thank you for coming out on a frigid morning to hear about a lot of really, if I'm going to be honest, a lot of really kind of scary, gross things. So, no? Can't hear you? Are we back? No? Coming? Slowly? There it is. Okay. If you have questions as we're going along, just wave. I teach full-time at St. Norbert, so I'm very used to people interrupting me saying, I don't know what you're talking about. I won't take it personally. Again, it's an unusual field. And I realize that for some people, microbiology can get a little dry and technical. I find that odd myself, but I've tried to put in a few little stories to that will make sense to anybody, even if you don't happen to find bacteria as interesting as I do. So the topic that we're talking about today is the human microbiome, which is something that it's a word that one of the good things about being a scientist, we get to make up our own words, right? The microbiome. We didn't use that word until just a few years ago. So I'm going to talk to you a little bit about what we know about bacteria, what the organisms that we're talking about are, the colonization of our body by things that are too small to see, and some of the implications of that, some of the ways that's changing how we're thinking about these organisms, and hopefully some practical applications, why we care, right? Which is always an interesting and difficult thing to keep going. So I'm going to start with a story about a collection of events that sort of got me looking at this field a little bit. That's me, by the way. I should mention that my primary work is on gliding motility in Flavobacterium. And I don't expect any of you to care at all about that. There's a group of us that get together. There's an international meeting for Flavobacterium biology every 2 years, and the same 80 of us get together and talk about this. But this is my colleague, Dr. David Bailey, who started at St. Norbert the same year I did. He's given at least one and possibly two lectures up here. He works in neuroscience and behavior and memory. And we talk a lot. People say we look alike. I don't see it, but that's fine. And we chat about biology and big ideas. And one of the things that came out while we were talking was this idea of a book came out called Microbial Endocrinology. And again, we're scientists, so we talk about these things a lot. I work with microorganisms, bacteria, single-celled tiny things. I'll show you some pictures very soon. Endocrinology is the study of hormones, how signals move around a great big body like ours, right? The idea that these two things are related seemed odd. Bacteria don't have hormones, they don't need them, right? But we were talking about how it would be interesting if we could merge the two things that we study. And we had a group of students that were in both of our courses who said, let's do some research on this, right? And I put the names of some of the students. Whenever I give a presentation of any kind, I try to put students up because it's what we do at St. Norbert, right? We get students in the lab, we get them doing research early, and this group of students who are all graduated now are at places like Purdue pursuing PhDs in cancer drug discovery, veterinary medicine, osteopathic medicine, medical degrees. I always tell people when you have fast players, you look like a good coach, right? So I'd like to take a lot of credit for this, but a monkey could have taught these kids. They're brilliant, right? So we got this group together, and they started looking at hormones. If you're familiar with hormones at all, has anyone heard of epinephrine? Okay. Or how about corticosterone? A little bit stranger with the glucocorticoids. Okay. These are stress hormones. Right? These are, we would call this the fight or flight kind of trigger. When you're startled or frightened, you get that surge. That's epinephrine, right? Corticosterone is a longer-term stress hormone. It has a lot of mostly bad effects, I think, when elevated for over a long period of time. I know I was at a physical with a physician a few years ago, and she asked me if I had any stress in my life. And I remember thinking, who says no to that, right? But these hormones are in different levels, and they elevate when the birds are stressed. It's one of the things that Dr. Bailey looks at. So we did this experiment in a model organism. I'm going to be talking about model organisms several times as we go through the talk, because they're important in how we do things in biology. They're experiments that we just can't do on people, right? So he works on this bird, the zebra finch. You can find this in a pet store. I should also say that people at St. Norbert are terribly confused because his name is David and my name is David. I work on zebra fish and he works on zebra finch. Maybe that's why they think we look alike. Anyway, so we have this bird, right? And he's doing experiments to this bird that cause it to be stressed and looking at how it affects their memory. And this bird, as you might imagine, has a GI tract, a gut, for lack of a better term. And in the intestines of this bird are a lot of bacteria, right? That's probably not surprising to most people. We know that bacteria live in poop, for lack of a better word, right? We'll try to skip some of the technical language. So we had students, and they have a very long and detailed protocol for collecting, essentially, bird poop and looking at what grows, right? What kind of bacteria can we grow in the lab on these little petri dishes, right? And there's a number of different organisms. And in microbiology, we spend a lot of time growing bacteria up, picking them off of these plates, seeing what that organism is, identifying it, right, and then doing experiments with it. And we did some of that. And then we had this idea, let's see if these bacteria respond to hormones. Now, there's no reason they should. They don't have genes for hormone receptors that we know of, right? They don't make these hormones that we know of. And I realize this probably is largely gibberish, but if you look at these different colored dots, these are different concentrations of hormones. And the text is small, and it's not terribly important to the point here. This is a bacterium that I bought from microbial shipping house. There's places, culture collections, where you can buy bacteria, right? This is a standard lab strain. The organism, Pseudomonas aeruginosa, you can get this off your shoe. It's a common soil organism, right? And if you look at these little colored dots, what you see is that they kind of overlap each other, right? This is measuring the organism's growth, right, over time. We watch it grow, and we treat it with these hormones, and this bacterium quite sensibly ignores them completely, right? These are hormones for vertebrates, and this is not a vertebrate. We'll talk about bacteria in a minute. When we took a very, very similar organism that we'd identified from the zebrafinch's gut, and we treated it with different concentrations of hormone, these lines separate, which means that the bacteria in the bird's gut grow faster when they detect epinephrine or corticosterone. And I'd be honest, I didn't believe it when the students brought these numbers in. I was like, sure they did. Uh-oh. That can't be good. Is that good? No. Kurt, any ideas? I'll hit return. Oh, I see. It's my... No, I don't see. Well, I'll explain what that means. Well, fix this. So I had the students do it again. They have argued that we put the re in research in my lab, because I thought, And we did it again, and we did it for 3 or 4 different organisms, and not all of them, but a significant chunk of them respond to hormones that the bird produces, right? Which essentially means they can tell when the bird is tense. Yeah, and it's exactly what I said. I was like, really? But apparently they can, right? The organisms know the bird's, I don't know how I'm going to say mood exactly, but they're responding to the same hormones that the bird is responding to. And if you think about it with a certain kind of eye, if you're a bacterium, we're going to show you some pictures in a minute, that bird is your world, right? That's your ecosystem. So I guess in a way it makes sense for you to know things aren't going well in birdland, right? Now what you're going to do about it as a tiny bacterium is something of a mystery, but so with that as a little bit of background, right? I want to talk a little bit about what kind of organisms we're looking at, right? What bacteria are. Oh, thank you. Is there anything I should do if that happens again? No, it went. Oh wait, that's because we need to get to this. Oh, it says low battery.
I decided to plug it in, so it's fine.
Okay. Where's the... This one?
Yeah.
I think that might do it. There we go. Okay, now we're back. Yeah, go on one more. There we go. Okay. So we're going to be talking today about this human microbiome. This is the cover of an article in the journal Nature. And if you're in my field, if you get on the cover of Nature, yeah, right, you send that one to your parents. That's huge. That's so what we have is, right, a person, and with, again, the right kind of eyes, what you see is not just a person, but an entire ecosystem of things that are not human, at least in the way we conventionally think about them. So we need to talk a little bit about microorganisms, right, which is what I do all the time, and you'll have to stop me if this starts getting too dry, because I could do this all day. When we talk about the microorganisms that we find, we're usually talking about one of 3 things. Bacteria, viruses, and fungi. There are other things that are classified as microorganisms, but these are the big ones. Fungi are eukaryotes. I'll show you a picture in a minute. They're reasonably closely related to us. They're just really small. Okay? Things like yeast and mold are fungi. Bacteria is where I'm going to spend most of my time, so I'll hold off on those for a minute. I want to talk briefly about viruses. We are host to a lot of viruses, right? Most of us have been hosts to a lot of viruses recently this winter, okay? Viruses are a different kind of creature. They're not cells. They're little tiny packets of genetic material. And I'm not going to spend much time talking about them. They are an important part of the host microbiome of any organism. We don't understand them as well as we understand the bacteria, and we don't understand the bacteria very well. So I'm going to leave the viruses to another time and hopefully show you some pictures of what we're looking at. If this is one of your cells, right, and your cells are small, right, this is a micrometer, okay, you can't see them, right, without a microscope. But if this is one of your cells, right, that's the nucleus. If you remember high school biology, you've probably seen these under a microscope. This is a typical bacterial cell, size-wise, right? Maybe a 20th or so the size of one of your cells. They're small, right? But I can see them under a conventional light microscope without much trouble. Viruses are even smaller. And one of the interesting things when you study microbiology, there are viruses that infect bacteria. You're never too small to have parasites, it seems, right? In fact, some of the viruses that we find in the human gut are predators, essentially, of the bacteria that live in the human gut. There's a group in Russia working on viruses as therapy, as a kind of anti-microbial, which works really well. It's just tricky to get people to take a virus to cure their condition, if that makes sense. So anyway, these are the organisms that we're talking about, right? We're going to spend most of our time here, and I'm going to tell you another quick story. Okay? When I first started as an assistant professor at Penn State campus, I I was asked to teach the first few weeks of a course in astrobiology. Anyone know what that is? It's the study of life on other planets. And I remember thinking, well, they can't prove me wrong. I can say whatever I want, right? We haven't found any so far. But I remember sitting down and typing up my first slide, which was, the title of my slide was The Origin and Definition of Life. And I had 10 slides, and I went, maybe this is going to take longer than I thought. That's a pretty big topic, right? One of the things about working in microorganisms is they're simple, so we have to get very simple in our thinking. And when you look at living creatures, right, and you ask a question that starts with why. Why is this organism doing what it's doing? And I don't care what it is, if it's a bird, if it's a plant, if it's a fungus, if it's a bacterium. If your question you're asking is why is it doing this particular thing, the answer is going to come down to one of two little arrows, right? Either it's doing what it's doing to get food and energy, right? That's what it's trying to get, is something to eat, right? Or it's doing this in order to make copies of itself, right? In very simple human terms, food and sex is why things get up and move around. Other than that, they just sit and watch TV, right? Oh dear, that's on camera, isn't it? Sorry. Anyway, what we talk about is what we call machine functions, things that cells do that don't happen normally, chemistry that shouldn't occur under the conditions. You're doing it right now. You're burning glucose at essentially room temperature, and that doesn't happen unless it's part of an organism, right? The other thing that all cells do is replicate. They store and transmit information from one generation to the next, right? Those are just core functions of living systems, right? All cells, and again, I don't care what the cell is from, will do these two things, okay? That helps us in our thinking about what the organisms that live in us are doing, right? We have known about bacteria for a very long time. Anton van Leeuwenhoek, late early 1700s, developed a very primitive microscope, but was very good with it, and was able to do drawings of a bunch of bacteria, right? That's mostly what he's looking at here. I love figure E. It's that circle. That's the organism, right? And that's what he saw, right? Many of these he saw from scraping his teeth, right, to look at. And these organisms are still good drawings, right? This is what we see when we look at a drop of rainwater, right? He didn't know what these were, but he saw them do things like move and said, well, they must be alive. They must be creatures of some kind. He called them animalcules, okay? And that brings us to one of our first sort of central points. Bacteria are diverse, really diverse, right? But they all look alike, pretty much, right? Under a microscope, you see spots. And these spots, right? Anything just jump out at you? Like, right? These are two organisms that I can find in my intestinal tract. I am more closely related to an oak tree outside than these two organisms are to each other, right? They are stunningly diverse, okay? And if you're my age or a little older, if you remember high school biology, who had the five-kingdom tree? The Plantae, Animalia, Plantae, Fungi, Protista, and Monera, I think it was. Look at that. I remembered something from 6th grade. Anyway, it turns out that that tree was wrong. When we take the organisms, all the organisms on the planet, and we measure them using the same scale, it's a genetic scale, I'll be talking a little bit about it later, what we find is a tree that looks like this. We have this common ancestor a long time ago. Here are animals, here are plants, here are fungi. Essentially everything you've ever seen is right there in that little corner, right? The rest of this is microbial, right? And these two are essentially kinds of bacteria. They're what we call prokaryote cells without nuclei. This whole mass, right, 90-something% of the organisms on the planet are in this one, what we would call this one little sliver, because they're small. We couldn't see them. We didn't know how to classify them, right? So it is a microbial world, right? I'll show you some better examples of that in a minute, right? And the length of these lines is a rough gauge of how closely related they are, right? So again, that's me, that's a tree, right? Here's one organism that lives in my intestine, here's another, right? There's a complete ecosystem living in my intestine that I don't have a clear understanding of. So, in addition to being incredibly diverse, bacteria are really, really old, like geological time old. My colleagues in geology tell me that the Earth settled around 4.6 billion years ago. Not too long after, in geological time, about 500 million years, right? We get the origin of cellular life. We see evidence of that. And this is microbial, right? From here all the way around to there, everything on the planet was a single-celled organism. And if you're looking again, this is the present, right? You follow this thing around. We start to get oxygen. Oxygen is the byproduct of bacteria, cyanobacteria, photosynthetic organisms that produce oxygen. There wasn't oxygen, free oxygen on the planet until somewhere around 1.5 billion years ago. And what we see is as we get closer to where we are now, we start seeing things like vascular plants and mammals. You know, if this was a clock, mammals appeared about 20 minutes ago. Okay? The fall of Rome was, well, it was since we had problems with the computer in that little window, right? We're talking about this geological time that is hard for our brains to process. It's just, right? And I'm going to come up with these some numbers again that I think are, we'll encounter this problem again. We're not designed to deal with billions, okay? But bacteria have been around for a very, very, very long time. This planet is a microbial world, right? We think we're the dominant species, but we're not. They were here long before we were, and they'll be here long after we're gone, okay? So, a couple of things I can tell you about bacteria. They are ubiquitous. You find these things literally everywhere. And I have a 9-year-old who's at the pool at the moment who said, and tell them that they're gross. I said, okay. Because they are. If I say, I want to find some bacteria, where do you suggest I look, right? Well, places like a dead fish, right? Or raw sewage. Okay? Can we find bacteria there? You bet, right? We can find them on the stage. I can find them on my hands. I can find them on my laser pointer. I can find them anywhere. Bacteria are literally every place. So while they're gross, they can also be beautiful, right? If, again, we look with the right kind of eyes. This is the water that dead fish was floating in, right? These are organisms isolated from that. But they're not isolated. It's a picture of lake water, right? This is a picture from raw sewage that's stained so that the different kinds of bacteria, different classifications, groups, I won't even call them species because they're not that narrow, are shown in different colors. And you've got green, you've got blue, you've got red, you've got probably 15 different kinds of bacteria in this little sample. And that brings up an important point that we're going to deal with later. When these organisms are out in their natural environment, they're living in communities, right? Just like we do, right? We live in a city, and there's, you know, butchers, bakers, and candlestick makers. There's all different kinds of us as we go through our day, right? When we study these organisms, we tend to take, you know, that one and pull it out by itself and then ask how it behaves, right? It probably behaves differently when it's with its friends, all right? So we're going to have to come back to that later. This is a lake in Spain that turned purple for a couple of weeks late in the summer. The color is due to certain photosynthetic bacteria that produce a lot of purple pigment. The entire thing was purple, perfectly safe. You could swim in it. No one would because they were afraid of them, right? Because there were bacteria in it. But again, lots and lots of different kinds of organisms, okay? And one of the things I find about bacteria is that they are surprising. They don't conform to our ideas of what living things are. This is Imperial Geyser in Yellowstone National Park. And if I told someone, go find me some living creatures, right, and I pointed here, I think most people, I'd go up there in the woods. That's where all the creatures are, right? This is boiling water. That's not where you look for living creatures, right? But it turns out this color that we see here is bacteria. The fact that it changes as we move away from the geyser and the water slowly cools is indicative of their different successional communities of microorganisms as the water temperature changes, right? There are bacteria that live, not just survive, but breed, actually reproduce in boiling water, which shouldn't happen, right? This is a boiling sulfur spring. And I was looking for an excuse to work this particular picture in because I love this picture. This is science in action. You can tell it's real science because there's duct tape holding this pole together, right? And this is boiling sulfur, pH of about 1. It would strip the skin off your hands even if it wasn't hot, but it's boiling. And this is a microbiologist. I haven't met him, but I think I saw him at a meeting once, who wanted a sample. So he taped a couple of bamboo poles together, right, with a little cup on the end, climbed partway down into the crater of the boiling sulfur spring, and asked one of his graduate students, well, just hold my belt, right? I'm confident this was not OSHA approved, but okay. It turns out there were living organisms in the boiling sulfur spring, right? That defies our idea of what life is, right? That's not how we think about things. The good news for us is these organisms live in boiling sulfur, right? I cool these things down to, 150 degrees Fahrenheit. They freeze to death, okay? So from a medical standpoint, I don't have to worry about those because I'm not made of sulfur and I'm not boiling, right? So it kind of restricts the pool that we deal with. But when we think about bacteria, what we mostly think about is that they're scary, right? When I say I'm a microbiologist, I watch people sort of wait and decide whether they should actually shake hands. Like, could be, you don't know, right? Because that's our experience. That's our most proximal experience with microorganisms. This is a picture of strep throat, right? Almost all of us have experienced that. This is a child with whooping cough, which is caused by Bordetella pertussis, a bacterium that we vaccinate against, right? This is a MRSA infection, methicillin-resistant Staph aureus, right, which you're going to see some pictures of later. That's scary, right? This is the interaction that we normally have with bacteria. This is how we think about them, right, when we get strep throat. And what do you do for strep throat? You take an antibiotic, right? And we're going to see some good and bad issues with that, right? These are legitimate concerns. And I'm going to be talking a lot about how we probably think too militaristically about bacteria and how we should treat them a little nicer than we do. But I want to be clear, if you have strep throat, take the antibiotic, right? This is an infection. This is bad. If you have a lesion like that, you need those bacteria to go away, right? The point that I'm hoping to make as we go through the talk is that it's not necessarily the organisms, it's that they're not where they're supposed to be, right? So we're going to be seeing that as we move along. The other thing that people don't like to think about as often is that bacteria are all over us. I mean, all over us. Most of us brushed our teeth this morning, right? And we do that every morning because every night we grow a layer of plaque, right? And again, having a 9-year-old helps with this because he's like, oh, is that slimy stuff I can scrape off? And I was like, yes, that's right? Because I didn't brush again today. I'm like, I know, right? Every one of these little dots is a bacterial cell. This is the surface of the tooth, right? These dots are bacterial cells. Between them is a carbohydrate slime, for lack of a better word, that they secrete. They produce a lot of that slime in response to sugar, sucrose specifically, the kind of sugar that we eat. So the more sugar you eat, the thicker and more slimy this layer gets, traps the bacterial waste products up against the enamel of your tooth and causes cavities, okay? This is an experiment that was done in England, actually, where they asked someone to just not brush your teeth. And then we're going to stain for bacterial growth. After one day, you start to see right at the tips of where the teeth meet the gums, as you would expect. After 10 days, it was 10 days, single guy, it turns out. Right? Who else is going to do this? Most of the tooth is covered with something we call a biofilm, right? It's this. And what's interesting about this, and this is I apologize, it's kind of a poor picture. The really dark spots here are a particular species of bacteria that we associate with tooth decay. These other spots are something else. They're bacteria, but they're not the one that we say, oh, that causes tooth decay. Streptococcus mutans, right? They're other organisms. There's an entire community living in this gross slime layer, right? So we're coated with these things, and we have to, again, by all means, brush your teeth, right? You don't want this on your teeth. Your teeth are unhealthy, okay? We do a lot of things to control how bacteria grow, right? A lot of traditional things. We wash all the time, right? We cook our food. We've been doing this for tens of thousands of years, long before we knew anything at all about what the organisms were. We knew if we didn't cook our food, we got sick, right? Refrigeration is essentially a way to make the bacteria that degrade our food grow slower. It doesn't kill them, it makes them slow down. You can do this experiment yourself. If you leave your milk in the fridge long enough, it goes bad. Who's done that? I've done that? Okay, yeah. We bury the dead or do something else. Now, there are lots of reasons why we do that, but one of the reasons is that we host a bunch of organisms that are very good at eating things like us, which, again, makes sense. Whenever there is a tremendous natural disaster and we have problems dealing with a large number of dead bodies, disease runs rampant, okay? There are a number of food preservation techniques. I'm married into a Scandinavian family, and one of my I guess it was like a rite of passage, I had to eat lutefisk. I mean, you really need to keep your fish if you're going to eat that, right? But fermenting, canning, all of these things are using particular activities to restrict the amount of growth of bacteria, because if we don't, right, they'll eat our food. Probably the most important thing that we do as civilized people is separate sewage from drinking water, right? And I want to make this point really clearly. The human population has expanded tremendously in the last 1,000 years or so. Part of that's due to agriculture. We've gotten much better at producing high-calorie foods, right? The other main thing is that we've gotten good at separating sewage from drinking water. We weren't very good at that for a long time. And the reason I want to make that point is we're going to talk a lot about bacteria that live in our GI tract and how we need them there, right? But we need them there. We don't need them other places, right? This is John Snow, an English physician, a very famous story of an outbreak of cholera in London in the 1860s. And he produced a map. You can go find this. Actually, Google Maps has done an updated version of where the cholera cases are. All these small black dots are cholera cases. And what Dr. Snow was able to show, and it took a lot of convincing because people didn't believe it at the time, was that cholera is transmitted by water, and it's transmitted by water that has been contaminated by sewage. Right? At the time in London, there were private water companies, and while none of us like to pay taxes, in some ways it's good to have a Department of Public Works that separates sewage from drinking water. I tell students all the time, the Department of Public Works does more to safeguard your health than the Department of Public Health. Right? If we don't do this, very little else matters. I don't know if you're familiar with cholera. It's a diarrheal-based disease. Patients with severe cholera can lose 20 liters a day from their large intestine. That's 5 gallons. I don't care what else happened that day, that's a bad day, right? That's just, that's not one you're going to want to repeat. Terrible, debilitating disease, right? And it's transmitted by water, okay? Water that's contaminated with sewage, right? So it's really important that we separate those things. So Dr. Snow did this sort of work in the 1860s, and a little bit later, a number of microbiologists, Louis Pasteur and a German microbiologist named Robert Koch, were working on isolating specific bacteria and saying, you know what, these cause diseases. And full disclosure, I was raised by a German Lutheran mother from a farm, and there's a sort of Germanic tendency. They like things to line up, right? We mowed the lawn on Saturday because it was Saturday, and that's when you mow the lawn, and we want things to be, the rows have to be straight, okay? And the reason I'm talking so much about myself will become clear later. We're talking about organisms. There really is a point to it. So what Robert Koch did was he wanted an orderly, efficient way to determine this particular organism causes this particular disease. And he came up with a set of rules, because he's German and that's what they do. And the rules were this. First of all, you have to find the organism in every case of the disease. Then you have to isolate it. You have to get it out of the sick person onto an artificial Petri plate in the lab. Grow it up so you know that's what you've got. Then you take a healthy individual and you infect them with it. And this caused a certain amount of consternation. He was working on anthrax in sheep. You can do that in sheep. You can't do that in humans, but we've worked around that with animal models. If you can take that bug, put it in someone, and get the disease again, see the same set of symptoms, and then, again, just because he's German, you have to re-isolate it from the new person to make double sure that that's really, really, really it. Then you have formally proven that this particular organism causes this particular disease. Does that make sense? Right? Very rational way to approach things. And I'm going to be talking about a lot of departures from this basic idea, but this has worked very well for us. Okay, I've got a couple of slides about that. If we look at what people die of in the United States, we start at 1900 and we go to 2000-ish. This red line represents death due to infectious disease. And you don't have to be particularly gifted at reading graphs to see that the basic trend of this line is down, right? We've done a really good job with infectious disease. Does anyone notice something odd about that red line? Yeah. I have students who are like, hey, what happened there? Right? Anyone know what that is? 1918? Influenza, right? This is, I know one of the great things about working with young people all the time, I say, what was happening in the world in 1918? And they kind of look at each other, and someone eventually goes, a war? Kind of, I was like, well, good guess. There's usually a war somewhere, but this is one of the big ones. This influenza outbreak actually killed more people than the war itself. So many that we don't really know, probably north of 50 million people worldwide, which at the time represented 1% of the human population. So we lost about 1% in 18 months because of the flu, which is one of the reasons that we spend a lot of time studying this particular flu strain, because this one's not like all the others, right? Flu makes us nervous because it can do this. Flu is a virus, by the way, right? Not a bacterium like we're going to be talking about. But right, this is good news, right? The sort of linear in-the-box thinking has really worked, looking at bacteria as pathogens and how we deal with this particular one that causes this particular disease. If we look at a little bit more detail at what people died of in 1900, right? The top three killers were respiratory diseases of influenza and pneumonia, tuberculosis, which we almost never hear about anymore, and gastrointestinal diseases, infections of the intestine that were almost always food or waterborne. Okay? Heart disease, stroke, those are down a little ways. And then down here, the really tragic one, infant diseases that are now almost entirely prevented by vaccines, measles, mumps, rubella, and diphtheria, which we really don't see much anymore. Okay? If we look in 2000 again, right? Influenza and pneumonia are still there. We have a new infectious disease, AIDS, which didn't exist in the human population in 1900, at least not outside of Central Africa. And heart disease, cancer, and stroke are way up at the top, right? We haven't done a very good job of making the potential human lifespan longer, right? That wall around 100 is still pretty much where it is, but we've done a very good job at helping more people get there, right? We've done a very good job at preventing infectious diseases that tend to take people early in life, okay? And again, we've done this using what I'm going to call the sort of traditional view of microorganisms as antagonists, things that we want to get rid of. My mother, who I mentioned before, was a nurse. She went through nurses training in the 1950s. And if you had a cough, you got penicillin, right? That's what we did, okay? And that's still what we do to some extent. Sort of adding, I guess, or sort of filling in with that antagonistic view of microorganisms is a look at the human body. And if you look at your body, it is designed, for lack of a better term, to not be eaten by bacteria. We're big antimicrobial machines, right? The design of your nasopharynx it seems random, but it's not. When you breathe air in, it bounces around all over the place so that particulates get stuck, right, in your nose. You blow your nose a lot now when it's cold, you get right when you're out breathing. That's on purpose, right? You catch a lot of particles that are coming in to keep them out of your lungs, right? There are cilia in your airway that push mucus up out of your lungs and dump it into your GI tract. Happily, that's down here below where you can taste things, so you never know that's happening. That's good, right? And those organisms are digested, right? So the lungs are very nearly sterile, even though they're exposed to the outside because of this. Tears, saliva, anything that kind of leaks out of you has an enzyme called lysozyme, whose purpose is to cause bacterial cells to pop. It digests their cell wall, right? Without that, your eyes get a lot of infections, okay? So we secrete this, right? We've got a whole immune system, right, that is designed to deal with bacteria that are floating around in your tissues, bacteria, viruses, that sort of thing, okay? I teach a course in immunology. We spend 15 weeks going through what happens when your body decides to get rid of a bacterial invader, right? And it's impressive. You've all had, right, inflammatory responses. You know what's going on, right? It completely reorders how your body's doing things. And it's extremely hostile to foreign invaders, right? Your stomach is very acidic. Not many organisms can survive that kind of acidity, right? A whole bunch of changes like that. We also flush and shed. Aside from your tooth enamel, every surface that is in contact with the outside world sheds. Skin cells fall off. There's mucus in all of your membranes. The cells in your mouth and throat, you eat a bag of Doritos, those scrape off by the thousands, right? And that's on purpose. We push those cells out, they get all colonized, and then we throw them away, right? And that's so that we're not constantly digested, right? And if you think for a minute, like, are these things really important? Think about a burn, right? Any kind of break in your skin that's larger than a little nick always gets infected. Burn patients have terrible trouble with Pseudomonas aeruginosa that I mentioned before, that's on your shoes right now and in your nose and all over the place. You encounter it all day, every day, and it's fine unless your skin's not there. If it gets in your muscle, it'll start eating it, right? And this gets back to that idea of all cells are trying to either eat or reproduce, and those two things are obviously linked. Bacteria, even the ones that cause disease, bacteria and viruses, they don't make us sick because they want to. They make us sick because we're tasty, right? We're a great big bag of proteins and carbohydrates and things that they like to eat, right? Nothing personal, which I know doesn't help, but there you go. The other two things I want to point out here are something we call normal flora, and that's what we're really talking about today. Any human organism, right, is completely, right, we have all of these defense mechanisms. And yet bacteria are still all over us. One of the running jokes in my microbiology class, I show this slide and I tell the students they'll want to memorize these names. And every once in a while, one of them takes me seriously. I feel really badly about that because the irony is missing. This is, to call this list non-exhaustive doesn't do it justice. This is just a casual list of a few of the organisms that we can easily culture from your skin, your mouth, your respiratory tract, urogenital tract, gastrointestinal tract. These live on us all the time, right? And we're sick without them. I'll show you a couple of examples here, right? This is another student of mine, a student researcher. She was mentioned before, Renee Wenig, and I always like to show this picture too, because she's sticking a swab in her nose. Very, very talented student. Straight A's throughout her college career, double major in biology and math. I don't know how you do that. I mean, I'm a professor, but I was like, wow. And also had the ability to convince perfect strangers to let her stick a swab in their nose without even blinking. And she did a little survey where she stuck this swab in her nose, went all over campus, the St. Norbert campus, healthy college students, and put them on a media that selects for the growth of Staph aureus in particular, right? You've heard of Staph aureus, maybe? Staph infections, MRSA, that nasty thing we saw before. And this yellow color here indicates that this person had staph in their nose, right? And then she tested them for antibiotic resistance. And I won't go through how we do that, but if there's no zone around this little disc, then they're not susceptible to the antibiotic. They're not susceptible to this one. They are susceptible to that one. It was this big experiment. She got 120 students, took swabs from their noses. About a third of them had Staph aureus in their nose. And when we look at the literature, that's typical, right? Now, can you get a staph infection? Absolutely, and they can be nasty, right? But a third of the people in this room probably have staph in your nose right now, and you're fine. Even more interesting, 5 of this 38 had strains that were resistant to all but one of the antibiotics that we tested them on. They were essentially walking around with MRSA in their nose. And we've looked again in literature in bigger schemes, and that's not atypical, right? So clearly, just the presence of that organism doesn't mean you're sick, right? And that's where it starts getting a little fuzzy. These bacteria are all over us, but not randomly. And that's where we're going to have to spend a little bit of time. If you take something like Staph aureus, and you can just swab it on your fingers. We can't do this anymore because of lawyers and things, but we used to just put it on people's hands. And you take E. coli, which is an organism that lives in your intestine, and you let them just sit on your fingers for half an hour. Don't do anything. Don't use Purell, don't wash, nothing. Just let them sit there. And then you press your fingers on media, little petri dishes, where the bacteria are happy to grow. The staph are still there. They're perfectly comfortable on your skin, right? Despite all of the different defense mechanisms your skin has, none of them work on staph, right? The E. coli are all dead, okay? So while your body is antimicrobial, it's not randomly so. It's not opposed to bacteria globally. It's opposed to certain bacteria in certain locations. Does that make a little sense? So the bacteria that we're going to talk about, we're going to spend a lot of time in the intestine. They're perfectly normal in your intestine, right? You have to have them in your intestine. But they're not normal in the urinary bladder, or in a cut, or in your eye. There, you can't have them, okay? It's like when I went to my grandmother's house. We could play in the basement, but there was the one room with the good china. You couldn't go there until you were a certain age, right? This is important in how we understand the organisms because we've tended to not treat them like this, right? If you think back to Koch's postulates, if you find the organism, we've got to deal with it. And that's probably not exactly true, right? Okay, so the other things that I'm going to kind of point out is that, again, I'm a parent, so if you're parents, these kids are healthy, right? We've done observational studies, and it doesn't matter what your demographics are, kids from every continent, every population, somewhere around the end of their first year, start to eat dirt. They pick it up and they chew it. I remember this. Took my kid to the beach, grab the sand, he's like, right? And you're like, and if they don't do that, excuse me, they tend to have problems later in life, which we'll talk about, right? Kids with multiple pets, right? Kids who live on farms have far fewer allergies than kids who live in cities. They have less asthma, right? They have less irritable bowel syndrome-based conditions, right? We need to be colonized by bacteria. just the right ones. And the fact that every kid in the world will eat dirt, right, probably tells us something that I'm going to come back to multiple times, and this is something that a long career studying biology has taught me. Nature usually knows what it's doing, right? It's important to intervene. Antibiotics are a wonderful, wonderful thing. They have saved countless lives, right? But if you look at the system globally, most of the time, your body knows what it needs to do if you just get out of its way, right? So we're going to come back to that. So these are healthy kids, right? But these mice are not healthy. These are mice that are being raised in a germ-free environment, and we can do this with mice. It's not easy, and these mice are, I think each one of these is about $8,000. I mean, these are really, really, oh my God. When I talk about money later, just close your eyes. These are really expensive experiments to do. But we can raise mice that are completely germ-free, okay, in these chambers. And, right, again, I had a mother who was a nurse in the 50s, and man, germ-free was what you wanted, right? But if you can actually attain germ-free status, these animals are sickly, right? They have very poor feed conversion, right? We give them mouse chow, they grow about a third slower than their germ-filled peers, right? They don't turn food into body mass very well. They tend to be a little bit lethargic, right? But when they do move, they're nervous, right? They're really susceptible to any disease that we allow them to be exposed to when they've grown up, right? So it's like their immune system hasn't had any practice, and so it doesn't know what to do when they encounter an actual pathogen. They are prone to asthma. They are prone to irritable bowel disease, right? And they have really interesting stress responses. If you put them in a situation where they're frightened, their response is much stronger than a normal germ-filled mouse. They're nervous, for lack of a better term. They do less exploring behavior in a new environment, but when they're in a comfortable environment, they're moving all the time, right? And they have some memory problems, okay? So because they don't have bugs, they're sickly and nervous, okay? And we're going to see a bunch of studies with mice as we go along. Mice are an imperfect model for gut bacteria for humans, but they're the best one that we've got, so we'll talk about them as we go along, right? So the sort of take-home at this point is that we have studied bacteria in a particular way, a sort of antagonistic way, for a long time, and that's not necessarily bad, but that there are other ways to think about them, right? Symbiosis is essentially two organisms living together, right? And there are really three ways that can go. There's mutualism, there's commensalism, and there's parasitism, right? Mutualism is when everyone benefits, right? I have a neighbor, another professor at St. Norbert, who also started when I did. He has a truck, pickup. He lives about 2 blocks away. I have a chainsaw. It's a mutualistic, right? He had 2 trees. I said, well, you know what? I'll bring him with the chainsaw. I can borrow your truck. Right? This works out for everybody wins, right? There are a bunch of bacteria that we know. There's bacteria in your gut that synthesize vitamin K. You can't make that yourself, right? So you can get it in the diet, but you don't get enough usually, right? So there are a number of cases where we know, we have nice, clear evidence that this particular organism and you are both getting something out of this. There is commensalism, right? Where you're not really getting anything out of this, but it's also not costing you anything. One person's getting something, one of the people. There is an organism called Corynebacterium that lives on the lens of your eye. It's okay, it's not a bad one. And it digests the little fatty acids that your eye, the supporting structures secrete to keep your eye wet. You were throwing those away anyway, right? This one doesn't cause infection. It doesn't try to get any deeper. It just lives on what you were throwing away. So that's fine. They're commensals, okay? Where we have probably spent the lion's share of our time is parasitism, right? Where one is benefiting at the expense of another. This is a roommate that doesn't pay rent. Right, this is, right? These are the ones that cause diseases, and that's, right, that gets your attention. I remember, again, back to being very small in an English class and hearing that a story has to have conflict or it's not a story, right? This is what draws your eye. And this is where we spend 90% of our research funding, is on the organisms that make us sick. And again, that makes perfect sense in a lot of ways. None of us want to be sick, right? So I'm going to end this little section of the talk, before we take an intermission, on what I call a tale of two posters. This last spring, I went to a meeting of the American Society for Microbiology. There is one. My very first ASM meeting was in Miami Beach in the late 90s. Great place for meeting, by the way. This is great as well, but the weather is a little better in Miami Beach. And when I was at Miami Beach, I remember this is my first big scientific meeting, and at that time, Miami Beach was populated mostly by retirees from New York City, a bunch of East Coast club kids that came down for the nightlife, and European fashion people, lots of really tall teenage girls that needed to eat some more, and then 9,000 microbiologists. And I'll be honest, we didn't blend. It's just, you could just kind of like, oh, there's one, oh, there's the black socks and sandals, there we are, right? So the convention center in Colorado, I don't know if any of you have been there, for reasons that are obscure to me, it looks like a giant blue bear is ready to leap through the window and devour everyone. This is a bad picture, I apologize. That's me, this is a research student of mine and some other research students. And we ended up taking two presentations to this meeting. One of them that Rachel was doing was this very classic, this organism that causes a particular disease in another organism, analysis of virulence factors, what about that bug lets it cause disease, how do we prevent it, straight up disease-based Koch's postulates microbiology. The other one that we took was that other experiment that I was talking about, how some of these organisms respond to host hormones, right? And I guess my point here is that this one drew lots of attention. Right? This one is easy to fund, right? This one, people are like, wow, that's weird, and move on, because it's just not as cool as like, ooh, I have a new drug that will cure this disease, right? I'm going to argue in the second part of this talk that we should focus a little more on this kind of thing, because it probably matters a lot, and we can focus on it now, and I'll talk a little bit about why that is when we come back. And I was told there should be an intermission. I think this is a good break point for what we're going to be talking about, because the next part is going to be talking about a big initiative by the NIH to look at what actually lives in and on us, and what those things are doing, both good and bad. And I'll be honest, there's some really scary data-heavy slides on that, so you want to take a break now and get up and move around for a minute. Oh, thank you. Are we okay? Am I live again? Oh, yes, apparently. Whoa. Okay. Okay, well, I hope everyone got a little caffeine and sugar to fortify you for the little bit here. I want to make sure I leave some time for questions, so I'm going to go through a couple of these fairly quickly. I'm going to skip over some of the methods and hit the high points. The methods, to be honest, are the parts that are interesting to people like me and almost no one else. So we'll give those a little short shift. In the last 5 years or so, NIH has funded a very big study to look at, something. Am I back? No? Wait, it's doing something now. It's waking up. There I am, okay. To look at what the organisms that live in and on us actually are, what they're doing, just in a normal, what they would classify as a healthy adult, right? What does it look like? Because we've never really looked in a systematic way before. And it's one of their figures from their website, sampled a whole bunch of different sites. I will walk through them very quickly. The project outline ended up with 242 healthy individuals in, I think, St. Louis and Houston. They picked a couple of cities that had big places to do this. 15 sites for males and 18 sites for females, because they had to have a few specific vaginal sites. And I'm going to go through a couple of numbers real quickly. A great number of samples. They used these techniques of 16S and genomic sequencing, which I'm going to skip over for the most part because they're really hard to understand. This number I wanted to point out ended up with 3.5 terabases of genetic sequence. So that's, let's see, there's kilo, mega, giga, and tera. That's 12 zeros. That's a number that's so big that it doesn't make any sense, right? And that's one of the central things that I wanted to point out as we're going through this. There's a lot that I'm going to answer when people ask me questions that we don't exactly know, because we're dealing with, I mean, these are NSA levels of data dumps, right? I mean, these are huge, massive data sets, and sometimes we don't really know what to do with them. It makes the visualization of the data, like presenting it to a brain, very difficult, right? So I will show you some pretty colored pictures that I think will help with that, but some of the stuff we're still sorting. We have the data, we're crunching it, as we say in the field now. So I'm going to spend about 30 seconds on this basic methodology. One of the problems that we've had for a long time is that a lot of the organisms that live in our gut, we don't know how to grow in the lab, right? Which means we can't do anything with them. And we don't really know what they are. So what the more recent studies have done is skip the step where we grow it, and they've started looking at just the genetic material, right? You've heard of DNA testing. It's on all of the detective shows. There are billboards in Milwaukee. You can use DNA testing to establish paternity and things like that. We've gotten very good at identifying things just by their DNA, right? And this is how we do it. It's a very convoluted process that involves a lot of molecular biology, which happily I can skip over. It's how we did this tree. We look at not what does the organism look like, but what genes does it have? And then we can place it very carefully on this tree. And this has been validated a lot of different ways. It's a really good technique, and it means that we don't have to get the bacteria to grow. We just take a sample and we look at what genes are there, and that tells us what was there when we took the sample. Okay? It's the same way we can process a crime scene. We don't need the person to be there. They left a little tissue, and we can tell who it was based on that, if that's a close enough analogy. I'm going to skip over this part. You get a thing that looks like this, and then you get this, except you get terabases of this, right? And okay, and then what we need to do this kind of work is truly massive computer support. Your brain doesn't do this very well, and neither does mine. But the computers are very good at it. And I know this because I once sequenced an entire genome of a bacterium. Well, I didn't. That's kind of my point. When we did this genome, there were 14 authors at 10 institutions from 4 different countries that put together one bacterial genome, right? Well, the project that we're looking at, they have 681 bacterial genomes, and so this is massive, $150 million project over a decade, right? To figure out what is living in us and what is it doing there. So, what have we learned? Okay? We went through all that pain in the first part of the talk to get to this part. So, the first thing that we learned, and we kind of suspected this, but we didn't really get quite the scale of it, is that the microbiome is large. We've detected more than 10,000 different bacterial species in that 242 people, right? That's a lot, right? And we've grown less than 10% of those in the lab, right? So the stuff that I was talking about before is great, right? I mean, E. coli, which we always talk about, that's our lab rat in microbiology. If someone says bacteria and doesn't otherwise specify, this is the one we're talking about. It's the one that you can find in your intestine. It makes up less than 1% of what lives in our colon. It's just one that grows in the conditions that we can work with in the lab, right? So lots and lots and lots of species, lots of genes. And I'm going to show you a couple of pictures that I think will kind of put this in perspective. Roughly, if we concentrated all these organisms together, 2.5 pounds for an average person, right? Which is, you know, a 200-pound person, it's about 1 or 2% of your body mass. But that's a lot of bacteria to be walking around with, okay? If we look at it on a cellular basis, right? This blue represents, I had a request not to laser point as much, so I'll just say blue represents human cells in your body right now. Right? The orange right there represents bacterial cells, right? Which means roughly 1 in 10 of the cells that are in your clothes right now are actually human cells that grew from a fertilized egg. The rest of them are bacteria and fungi that have colonized you over your life. Okay? So we're not individuals, we're not organisms, we're ecosystems, right? To be a human means to be a community, right? Which is great. I work at St. Norbert College. We talk about communio all the time. I don't know if this is what they mean exactly, right? But we're not individuals, not really. The boundary between us and the rest of the world is not nearly as clear as we would like it to be, right? We bleed into the environment. And if we look at it in terms of genes, a few years ago we sequenced the human genome, and a really interesting talk on that, but that's a different talk. About 23,000, depends on how you count them, genes that code for what it takes to make a human, right? If we look at the bacterial genes, there's more than 1 million. There's possibly as many as 8 million, right? Which means roughly 1 in 300 of the genes that you're carrying around are actually yours. The rest of them belong to bacteria. A lot of those are redundant. A lot of those have nothing to do with you, and it doesn't really matter. But some of them you actually need, okay? And this is important because the bacteria are not evenly distributed, right? They're not everywhere. If we look at, and I'm not going to even go through what these names mean, right? Trust me, you don't want to know. Just look at the colors, okay? If you look at some place like the colon, right, which is here, there's a whole bunch of these bacteria that are red, the lactobacillus, right? There's a whole bunch of these that are Bacteroides. We only find them there, right? We don't find those almost at all on the skin, right? That purple one, right? We find almost nothing in the stomach except for a bacterium that is associated with ulcers, right? Which is another story in its own right. If we look at vaginal flora, almost all one kind of bacteria, right? And that matters a lot, and I'll show you why in a few minutes, right? If we look at skin, right, a completely different group of bacteria than we find in the colon or in the stomach or in the vagina. Very, very different populations. And if you think about it, it makes sense, because think of how small the bacteria are, right? If you look at a map of the United States and where people live, right? I recently drove to Fargo, North Dakota, long story. In Minneapolis, I was in one place, population density-wise. Between Minneapolis and Fargo, a very different place, right? There was not a lot, to be honest, right? So it shouldn't surprise us, I guess, that there are different populations in different places, right? But they're profoundly different, okay? And they kind of need to be. And when I say unevenly distributed, I mean really, really unevenly distributed. I want you to focus your attention just for a second on this and on that. Those are two microbiomes that were found on your feet. This one is on your heel, mostly red, right, which is Firmicutes, and a little bit of the green, which is proteobacteria. This is between your toes. Right? They're separated by about that much, and completely different ecosystems. It's like going from a rainforest to a desert, right? And they're inches apart, okay? So when we talk about bacteria on the body and in the body, we need to be really clear about where we are, because they're not the same, right? If I have an infection in a wound on my arm, that's one set of problems. That same infection is going to encounter a very different neighborhood if it's in my intestine or in my eye or behind my ear, okay? So we've been thinking about these things as one giant mass because that's kind of how we approach them, but they're not, right? They're very organized communities. And this organization is stable over long periods of time. If I take bacteria from the gut and I put them on the skin, they don't last there. If I take bacteria from the skin and I put them in the eye, they don't stay, right? This organization is not random, but it's also fairly stable, okay? And it varies between you and me and other individuals, right? These bars each one of these, the HVs, right, are individual people. Okay, human volunteer is what that stands for. And if you look, again, this is a skin of the arm here, I think. This is skin on the back. This is the heel. This is the nose. And if you look, you see the same colors, but not in the same ratios. Right? There's more green here. Number 3 is different from most of the others, right? There's more green here. We've got a lot of purple on this one, but not so much on that one, right? It varies. Mine is not the same as yours, which is one of the many reasons why some people will respond to a particular treatment with antibiotics differently than someone else, right? Because we don't all have exactly the same set of bugs, okay? But, and this is the important thing about looking at the whole genomes, the top graph shows variability in actual species, things that we can identify, Staph aureus or Pseudomonas aeruginosa or E. coli or any of those other bugs that we looked at. The bottom graph, this one, shows variations in genes that carry out different functions, and I realize that's kind of messy, right? But if you can just look at the two graphs and see that the top one varies a lot, right? It's not flat, and the bottom one doesn't. What that means is while my bacteria might be different than yours, I've got somebody who's carrying out the same essential functions as you do. And you can think about ecosystems on a bigger scale, right? Somebody is the top-end predator. If it's in North America, it could be a wolf or a bear or a cougar. If it's in India, it'll be a tiger, right? But somebody is occupying that role. Does that make a little bit of sense? So even though my particular organisms might be different than yours, we have roughly similar activities in a lot of ways, okay? And there are a whole bunch of things that affect how this works, right? Why mine might be different than yours. And I'm just going to go through a few of them, and then I'm going to talk about some of the functions that we know about, right? The first thing is how we enter the world. And this matters, right? If we look at babies that were born vaginally and compare them to babies that were born by cesarean section, they are colonized very differently. And in a follow-up to this, if you look at like these that were born vaginally, have a lot more of a particular kind of bacteria that we find in the vagina. And what's particularly interesting about this from a biology standpoint is during the last 3 weeks or so of pregnancy, the concentration of that particular organism in the vaginal mucus increases. As the mother is getting ready to deliver, she starts essentially feeding a different species of bacteria so that when the baby passes the birth canal, it's colonized by the right organisms. Now, who would have thought of that, right? We do a lot of cesarean sections. Now, some of them we absolutely have to. That's the, right? It's got to be done, right? But what we didn't really take into account was that that has long-term effects. It changes the founding population of the infant's skin and GI tract. And as we're doing some follow-up studies, the babies that were born by cesarean section do have a statistically higher chance of having asthma, irritable bowel syndrome, and some of the other diseases that we tend to associate with our immune system. We're going to get to cause and effect later, right? So I don't want to oversell that. But we do notice later on, even when you look at these kids as 4-year-olds, their intestinal bacteria aren't exactly the same, right? There's a little bit of a shift because of how they came into the world, right? And our bacteria change as we age, right? When we are young, when we are babies, if we are being fed, we're breastfed, we have a particular population that is different than if we're fed formula. There are sugars in breast milk that are undigestible to humans. We don't have the enzymes to break them down. But there are bacteria that colonize the infant gut that do have the enzymes to break them down. And I have been a long-term champion of breastfeeding from an immunological standpoint. There's all kinds of really important immunological molecules that you get from breastfeeding that you don't any other way. But this is something that we didn't think of. in addition to actually feeding the infant, it looks like, and I want to be clear that these are still suggestive, it looks like the breast milk is also acting as what we call a prebiotic. It's feeding a particular population of bacteria that are good for the infant digestive system, right? And that's kind of amazing when you think about it from a coevolution standpoint, right? That we have evolved to provide food for bacteria that we want in our infant's GI tract, right? As I said before, nature's pretty impressive when you are paying attention, right? As we get older, right around 3 years, when we start eating solid food, the infant bacteria in the gut change to look like grown-up bacteria very quickly, unless they're malnourished. And this brings up another interesting point. Obviously, if you're starving, the bacteria in your GI tract are going to have a different experience than if you're not, right? And they're going to be smaller numbers, they're going to be different populations. What we didn't understand until fairly recently is if you take a child that was malnourished and give them a good high-calorie diet, right, these tend to still not grow as quickly, right? They tend to not thrive as well as children who had never been starved. And if you look at the bacteria in their gut 6 months or so into a good diet, they're still not the same as bacteria in a gut of a kid who never was malnourished. So one of the things that some of the non-government organizations and people who are doing food aid are looking at is, do we need to add something to the emergency food pellets that we're giving people that are going to feed the bacteria that we need for them to develop in their gut to process this new diet, right? Is that something that we just didn't look at before, okay? When we get older, we have a microbiome. If we are overweight, which a great number of us in America are, the bacteria in our gut are different. And that's not surprising, right? I'm going to show a slide later, though, that suggests, at least in mice, if I take the bacteria from the gut of a mouse that's heavy and put it in a mouse that's not, the mouse that's lean will start to gain weight, even if I don't change its diet, right? So if you're trying to lose weight, blame your bacteria, I think. That's my excuse. I'm a microbiologist. I get to say that, right? Now, as we age, the microbiome changes as well. There's some profound, I got an email this morning from one of my research students who found an article while he was doing something else. Says, oh, you can use this for your talk, right? About how the bacteria change as we're aging, and that has an effect on how our digestive tract works. And again, we're going to get to cause and effect in a few minutes. What we eat obviously matters. If you think about what's going to live in your GI tract. There was a great big study, part of this NIH trial, looking at the diets of people in the US, right? In blue, people, the colors and the dots that are red are people in sub-Saharan Africa, a group of people, and in green is a group of essentially tribal people in Central South America. And you don't, again, have to be particularly quick on the uptake to notice that the American bacteria cluster separately than the ones from Africa or South America. And again, these are people on separate continents, right? But what they have is a gut microbiome that is more specialized to digesting carbohydrates. Their diet is largely corn and plant-based, corn and rice and plant, and that's what their GI tract bacteria are designed to eat. Our diet is higher in protein and fat, and we have bacteria that are good at eating those, right? Not, again, surprising. My 9-year-old wanted me to mention that our American microflora looks a lot like the gut bacteria that we would find in carnivores. We've looked at dogs, we've looked at wolves, we've looked at cats, and we see a lot more overlap in carnivores with this, humans that have a Western diet, and more with herbivores, not ruminants, but other herbivores, some of the plant-only eaters in these other diets. So what we eat matters, right, to what colonizes our body. So lots of variation, right, but an individual's microbiome is fairly stable over time. It does change with age like everything else, but The bacteria that live in your gut are probably pretty similar to the ones that were there 3 years ago. And they're probably going to be pretty similar 3 years from now. Okay? And we're going to talk about the gut. It's right after the holidays, so I picked this picture. And I've been in Door County for a while. You guys have good food up here. I gotta go back. This is crazy. Obviously, when we look at the GI tract, right, when we look at where we find bacteria, right, we're talking about in the colon between 10 to the 12th and 10 to the 14th cells per gram, right? If we take colon contents, we take a piece the size of the top knuckle of my thumb, there's 100 billion bacteria there, right? It's an inconceivable number, okay? Most of our organisms that we carry around are there. And if we look at that, when we look at some of the activities, it's kind of like we've been ignoring an organ, right? We have heart and lungs and liver and all the sort of stuff that you study in anatomy class, but we really haven't looked at what these things in our gut are doing because they're, let's be honest, it's poop, it's gross, we don't want to mess with it, right? And they're not us, right? But they're there all the time, right? They're processing our food as we are. They probably have some profound impacts on our health. And it turns out they do. A lot of the foods that we eat routinely, both plant and animal, we cannot digest by ourselves. We don't have the necessary enzymes to do some of the heavy lifting, breaking big plant matter into small plant matter, right? Once it's small, we can chew it up and absorb it, right? But when it's big, we can't. We rely on bacteria in our gut to do that for us. They're part of our digestive system, okay? Which is one of the reasons we think that the mice that don't have bacteria grow really badly. We've known this in cattle for a long time. People at Madison have been studying this for a while, that we have to have the right mix. Cattle, by the way, can't digest grass. They carry around microorganisms that can digest grass. They're big walking fermenters, right? But it turns out that our GI tract has important, or the bacteria in it are important in how our GI tract functions. And it's probable, although not certain, that we're feeding some of them. Our intestinal cells produce mucus. The components of that seem to be feeding a certain couple of populations of bacteria, Bacteroides and Bifidobacterium, if you're a microbiologist, right? So they're not really separate from us. They're part of our GI tract. And what are they doing there, right? Well, they're helping us digest food. They are also implicated, is about as far as I will go, in obesity and diabetes. Obviously, obesity involves taking in more calories than you burn up. The math there is pretty solid, right? But we all know people. I have a colleague, Dr. Fire, who, when we have an event on the campus, will eat 3 pieces of cake, and he's skinny as a rail, and always has been, right? Part of that's his own genetic makeup and metabolism, but part of it is probably what lives in his GI tract, right? When we look at the GI tract bacteria of people who are obese and compare it to people who are not, they're not the same. And that's not that surprising, right? We would think, you know, if you're eating a lot and you're eating more high-fat food, you're going to select for different bacteria. But when we look at some mouse studies, what we notice is if we take mice that are lean and mice that are obese, right, and we swap their gut bacteria and keep the food the same, right? All we do is we take the bacteria from the obese mouse and put it in a lean mouse. It'll start to gain weight even eating the same feed as it was eating before. And vice versa. If we take the bacteria from the lean mice, it will start to lose weight. And then, interestingly, the bacteria will shift back to the population that it was before when it was obese, and the mouse will gain its weight back very quickly, which is probably one of the reasons why it's really easy to lose weight for a couple of weeks, and then it comes back when you're not paying attention, right? It's not just your fat cells that are doing that. The bacteria probably reconstitute how they were before, right? And again, I mentioned the germ-free mice before. So we've looked at a number of different ways that the bacteria in the gut affect how we absorb nutrients, right? And I don't want to say that this is the cure for obesity, right? Obesity, it also involves eating less and exercising and all kinds of ridiculous notions like that. But it's probable or almost certain, right, that what kind of bacteria you have in your gut predispose you to gaining fat versus lean muscle mass. And that is something that we can probably adjust. And we'll get to that in a couple of minutes. The weirdest thing that we found is the effect of these organisms on cognitive functions, stress and mood. Now, we all know that when we're nervous, our GI tract is funny, right? We get butterflies in our stomach. We've talked about this for years, right? Before a big talk, you get nervous and like, oh goodness, I didn't eat much breakfast today because I was, afraid I'd forget my pants or something stupid, right? I wasn't worried about that really, but it's an example. But it turns out that many of the messengers, and this kind of goes back to that study that I was talking about before, some of the chemical messengers that our brain and nervous system use to send signals are also used by bacteria to signal amongst themselves. So when you get tense, it's not surprising that some of those chemicals are going to leak through your intestinal lining, and the bacteria are going to sense them. Okay? And some of the things that bacteria produce as part of their metabolism, it turns out, look an awful lot like neurotransmitters. Now, we don't know exactly yet, I don't want to oversell this, if those are functional, but they sure look like they would be, right? And I mentioned this before with germ-free mice, right? They're nervous, okay? Even when we raise germy mice in that same weird kind of environment, right? They are less easily the germ-free mice are more easily startled. They're more twitchy in their movements, right? And they have problems developing new memories. Okay, so we're confident that there are some effects on stress and mood. There was a big study recently looking at the gut bacteria of people with autism spectrum disorder. We've known for a long time that there's a GI tract implication there. Most people who experience symptoms of autism also have digestive problems, right? And some immunological problems. And it turns out that there are really clear associations, and in a mouse model of autism, we can reduce autism symptoms by changing what bacteria live in the mouse's gut. Now, again, I don't want to oversell that. That's in a mouse, and we're trying to project autistic-like symptoms on a mouse, right, which is a difficult thing to do, but they are social animals, and it's opened up some new areas of research, right? Okay, I've got a big table of things that we look at in the GI tract that are linked to human diseases, right? And I don't want to spend much time on it, but there's a bunch of them, right? I mentioned obesity, inflammatory bowel disease. I will show you a quick picture, right? If you just look over here at these bars, right? People with Crohn's disease have less of a certain type of bacteria in their gut. Is that cause or effect is something we're still working out, right? Again, some of the mouse models, if we fix this defect, the symptoms go down. And there's been good studies on using probiotics, things that change the microflora a little bit, in people with Crohn's disease to reduce symptoms. Doesn't fix the problem, but it makes it less severe, okay? And this is where we get to cause and effect issues. We have a profound problem now with cause and effect in the microbiome, right? This is a classic study that you can very clearly link shark attacks to ice cream sales at any beach on the planet. Ice cream sales peak in July, and so do shark attacks. Therefore, right, it's to remind us not to think that just because two things happen at the same time that one causes the other, okay? Our traditional way of looking at bacteria relies very much on presence or absence. Is Vibrio cholera in the GI tract, then you have cholera. Presence-absence is easy, right? What we're looking at is disrupted ratios. Is the percentage of Firmicutes compared to the percentage of Bacteroides the same as a healthy person, or is it skewed one way or the other? That's a much harder nut to crack. Finding something somewhere is one thing. Determining the ratio of that thing to a bunch of other things in a complicated system is really, really hard, right? I'm just saying that off the front. So we talk about something called dysbiosis, which is your bacteria aren't in proper balance. Okay, that's an unsatisfying diagnosis. You don't want that from the doctor. You want to hear, you've got this, here's a pill, right? This is a much trickier little system. When we talk about practical applications of what we have learned from the microbiome, there are a few. There are probiotics and prebiotics. These are ingesting either live bacteria. If you eat yogurt, you're eating bacteria like crazy, active yogurt cultures. They're lactobacillus, they're one of your GI tract bacteria. They're also vaginal. Very common bacteria in the body. They're actually healthy for you, right? We also talk about prebiotics, eating things that encourage the growth of bacteria that we like in our GI tract and discouraging the growth of things that we don't, okay? The problem at the moment is we don't really know what good prebiotics and probiotics are. Not really. We have some suggestions, but we don't really have a good handle on if you eat this, your bacteria are going to be in a better place, right? Partly because we don't understand why mine vary compared to yours and what exactly they're doing there. The other thing that I had to bring up in a human microbiome talk is the idea of fecal transplants. And I wish I was making this up, but I'm not. Brace yourselves. It's almost lunchtime. Okay. There is an infection that is growing by leaps and bounds, especially among people above 65, called Clostridium difficile. My mother actually had it. If you've ever been on a hospital, you don't have to go in the room. If you go on the floor of a hospital where someone has Clostridium difficile, you know. It's a GI tract infection, really nasty bug. It's caused by Clostridium. This is normal flora. We find this in the GI tract, this particular organism, of almost everybody, if we look, right? So lots and lots of people have this, right? But if we take a lot of antibiotics, and again, if you get an infection and you need an antibiotic, by all means take it. But if we take antibiotics and we take them again and we take them again, what we end up doing is killing off a lot of the bacteria that normally live in the intestine. I liken it to your lawn. If you plow up the grass, something's going to grow there, right? So if we get rid of what normally lives there, Clostridium difficile is really hard to kill. So it waits till everyone else is dead, and then it comes out and grows like crazy and causes terrible lesions on the inside of the intestine, massive gas production. It's a miserable condition. If any of you have had it, I'm sorry, I've worked through a few of these. And what happens is, right, we get this bloom of the organism, and because it's really resistant to a whole host of antibiotics, it's really, really hard to get rid of. And because what caused it, for lack of a better term, is killing everything else off with antibiotics, the treatment, right, just makes it worse. So we have this intractable problem. Well, how do we solve that? What someone thought was, well, when people have a healthy microbiome, they don't get this. So why don't we take, just like any other organ transplant, we'll take the gut bacteria from one of their relatives and give it to them. And it sounds really fine when you say it's an organ transplant. And there's this nice picture, look, see? But that's not what it really looks like. That's what it really looks like. And you transport it just how you think it's an enema. Again, I wish I was making this up, but I'm not. This guy, Dr. Liu, has developed a capsule so you can just eat this one. And people are like, I'm good, right? And it turns out, they've done a couple of trials. They did 77 patients at the Cleveland Clinic, 91% cure rate in 2 days for people who'd had Clostridium difficile for more than a month. They took the pill, and 2 days later, they were fine, right? There were 8 people that didn't respond. 7 of them, they tried it again, and it worked the second time. So profoundly effective therapy for a really intractable problem, low cost, just really, really gross, okay? So, in the interest of time, I will wrap this part up. Are we gracious hosts? I would say yes, right? We do all kinds of things to support the growth of bacteria in and on us, right? We inoculate our infants as they come past through the birth canal, right? We get everything ready. We feed them, right? So that the ones that we want to grow, grow, right? And I haven't covered this at all, but our immune system, which is really good at getting rid of things we don't like, has ways that we understand poorly of ignoring certain bacteria. I liken it to the cops in my hometown when I was growing up. there were laws, and they were enforced unless they kind of knew you and they knew that you were just being an idiot, and then they'd send you home and call your dad rather than take you in. It happened to a friend of mine, not that I have any personal experience with that, right? But our immune system understands more clearly than our brains in some way that not all of these things are bad, and it will ignore some organisms. It'll just say, no, that one's not a problem. And we don't understand clearly how it decides that, right? We're as sure as we can be, although we don't understand the nature of the communication, that we and our bacteria talk to one another chemically. We don't know exactly what we're saying, but we're pretty sure that we do. So are we gracious hosts? Yes, but we should probably be a little more careful with our guests, right? We tend to think of bacteria all as one big thing, and they're not, right? Some of them we need to bleach, because they'll make us sick, right? By all means, wash your hands and use the hand sanitizer. It keeps a lot of viruses and some of the nasty bacteria off of you, right? But we probably don't need triclosan in every product in our house, right? Because we're killing bacteria that we don't need to kill, and something else is going to grow there, right? We probably take way more antibiotics than we need, and I know we give more than we need to our farm animals, which is a whole other topic that I could talk about someday if you'd like. And what we need to do is get better at developing antibiotics that kill the bacteria we need dead and leave the other ones alone. Okay? We have a lot of what we would call collateral damage if we were military, right? We're killing things that aren't a problem. Okay? So what do we not know yet? First of all, what we don't know in a lot of cases is cause and effect. We see the changes at the same time. We know some from adoptive transfer studies, right? But we're really working out cause and effect, how our genes and their genes interact, and that is going to be a complicated bit of work, right? We don't know how malleable it is. How easy is it to change the microbiome, right? We know we can change it. We know it recovers from antibiotics, and then sometimes it doesn't, and we don't clearly understand what the difference is between those, right? We don't know what are good pro and prebiotics, not really. We have some inklings. We know that like yogurt is probably good for you, and bacon, as well tasty as it is, probably not, but we would like to firm that up a little bit. One thing that we're looking at, a number of people are looking at, is do we have microbial fingerprints? There's a group in China that just released a study that said they can predict who's going to develop type 2 diabetes based on their gut bacteria. And that would be really nice to know, right? We don't know how much of our flora is inherited from our parents and how much we get moving through the world, right? And we don't really know very clearly what our use of antibiotics does to the biome, because we've only been using those for about 60 years. Right? And we never really looked. We just assumed all bacteria were bad, right? So there's a lot of work to do. A lot we don't know. I'm glad NIH is doing this, right? And some of the work is really, really hard. But we're at a place, in a lot of ways, where our handling of infectious diseases is pretty good. The problems that we're having now, obesity, diabetes, autoimmune conditions, irritable bowel syndrome, these more chronic intractable things, because we're pretty good at the nasty infections that Robert Koch was working on, we can now start to work on some of those. Those are going to require a different mindset, right? They're going to require, right, a more, right, what's in here is a community. It's a jungle, right? We need to sort of tend it like a garden rather than clear cut. And this recapitulates what we've learned about the world in general, right? 100 years ago, we went in and cut things down because that's what we needed to do to build. Now we're kind of going, ooh, how about we leave some of those trees? We're going to need those later, right? We probably need to think less antagonistically. These organisms are part of us. They're not some weird thing that we need to get rid of. They're part of what we are, and they always have been. And I want to come back to the idea that nature is smarter than we give it credit for. I remember back in the 50s, this was a big thing. My mother used to do this. When we get an infection, iron levels, free iron levels in the blood drop. Well, we got to fix that. So we would give people iron, right? It turns out that restricting iron is a really good way to keep nasty bacteria from growing in the blood. Your immune system pulls the iron out of the blood on purpose, right? That's why we get fevers. There's a big movement to like, you know, if kids get fever and it's not too high, let them. They do that for a reason, right? So we probably need to start thinking a little bit more in terms of the body knows what it's doing, right? Only intervene if you have a better idea, I guess is what I would say. And with that, I would like to thank a whole bunch of people. I feel like I'm at the Oscars now, right? I have this long list, and they're going to start playing music in a minute. Obviously, I would like to thank the Door County Auditorium. This has been a fabulous opportunity for me to read a whole bunch of papers that I would not have necessarily otherwise read, and I get to talk to a whole bunch of people about bacteria, which I could do all day long. And obviously the college. The American Society for Microbiology has been very useful in letting me steal a lot of images from them. And many, many excellent students. I keep coming back to the fact that pretty much everything I know about this is because some student asked me a question and I realized, I went to grad school all those years, I don't know this, right? And we figure it out. So with that, I will stop talking and take any questions that anyone has. I'll be happy to sit on the end of the stage and chat for the rest of the day if you want to. So thank you for your attention, and stay warm. I might be running over a little bit. I do this all the time.
I'm just wondering if there's been any correlation between if it is in fact an increase in colon cancer and other kinds of cancer related to the digestive system.
There is a particular set, and I kind of skimmed over that in states of time. There are a particular set of organisms that are overrepresented in patients with colon cancer, and some of the products that they're producing are reactive oxygen species, cause oxidative damage to the mucosa. And again, in mice, we've been able to take those bacteria, transfer them to an organism, to a set of organisms, and their rates of colon cancer increase. So yes, the short answer is yes, there is a correlation between what's living in your GI tract and colon cancer. The longer answer is we don't quite know what it is yet.
I'm assuming there's a relationship between bacteria and pH level in the body?
Yes, so your stomach is pH around 2. Most people's stomach is, I won't call it sterile, but it's pretty close. Very few things live in the stomach. Things will pass it in food. They hide in what you eat to get to your intestine, because not everything gets broken down in the stomach. There is an organism called Helicobacter pylori, which will colonize your stomach. If you have this organism, you're more likely to get ulcers, because it survives that kind of pH. It burrows into your mucosa. Turns out if you don't have this organism, you're at a slightly higher risk of asthma for reasons that we don't understand at all. But yeah, pH is a huge, and I didn't go through all the details. When girls start puberty, they change the sugars that they produce in the mucus of the vagina that supports lactobacillus, which drops the pH to less than 5, and that prevents the growth of fungi. One of the easiest ways to get a vaginal yeast infection is to take an antibiotic that kills off the lactobacillus. It's almost like clockwork. So pH is a huge component of how we regulate the flora. Yeah. Other questions? Okay. If any of you have questions that you would rather ask in a more small setting, I'm happy to stick around for a while. I know some of these topics are like, did he just say that?
Thank you, Dr. Hunnicutt. Oh, maybe one more question.
Okay.
And thanks to all of you for coming out today. We hope to see you back here next Saturday. One more question.
I'm not sure how to ask this, but if someone has mycoplasms,
yes,
and can't seem to get rid of it,
any
suggestions?
Mycoplasm is a tough one. It's a very unusual bacterium. It's really hard to grow. Not easy to detect either. There's a cluster of these. And we don't know exactly how, but it seems to go dormant for periods of years. I've got one living in my left lung, I'm pretty sure. Well, I used to. Any suggestions? Not really, other than be generally healthy. Most of the people that I know that have had mycoplasma infections, and I'm one of them, full disclosure, the first one was right after I defended my master's thesis, and the second one was right after I turned in my dissertation, and I went 52 hours without sleep, and that turns out to be a bad idea. It seems to be a disease associated with stress, but it responds to antibiotics, but it seems that the antibiotics knock it almost out. There seem to be survival forms, and that is an active area of research. We can do some things in the lab, but as far as in an individual, I don't think we know that, or at least I don't. Wish I had better information, but that's about what I've got. So. Oh, yeah.
If you have a microbiome that's malleable,
Mm-hmm.
and you use a treatment like fecal transplant,
Mm-hmm.
does that mean that you will get a permanent result? Or will
Yeah.
it be-
Yeah, it turns out that when you do the fecal transplant, it recolonizes, and again, it self-organizes. The organisms that are in the small intestine, even if, and again, I apologize, there it is. It comes up, right, it's an enema. It colonizes the large intestine first, and they've actually done a couple of trials where they've colonized the small intestine, which is where much of the problem is, and the community will organize over a period of weeks. Bugs that like the large intestine will grow there, and bugs that like the small intestine will migrate up and grow there. It rebuilds a new microbiome, and it's permanent. Yeah. Yes, sir.
I wonder if you would address a common experience.
Mm-hmm.
You go from one culture or one location
Yeah.
to another location, and you experience a mild form of Montezuma's
Yes. GI tract.
Right.
Issues, as we call it.
How much of that is E. coli, and how much of that is simply different populations of bacteria
Yeah.
confronting each other?
It's a really good question. We know that you get used to the strains of E. coli. There are E. coli, by the way, that are really nasty, right? Those aren't the ones that normally colonize your gut. The O157H7 is a really nasty bug. It has some genes that are different from the common E. coli that we carry around with us. When you go somewhere else, you get a new population. And we don't like to talk about it, but you always get a new population, and it's always gets the same. It's called the fecal-oral route of infection, and no one likes to talk about that, but happens all the time. So some of those your immune system hasn't seen before and doesn't know what to do with. So at least part of the discomfort you experience is due to what we would call a classical infection. Those bacteria burrow into your intestinal mucus and secrete some things that make your GI tract unhappy. But part of it is a shift in diet. And we've done some experiments with this where we'll just have people, we'll just change the diet, right? We'll just like, rather than actually send them to Mexico, we'll just feed a Mexican diet for a couple of weeks. And most people do experience some level of GI tract symptoms. That's probably due to their bacteria readjusting, because now the food's different, the water's different, everything is different, and it's sort of like getting over jet lag. Your bacteria have to reshuffle. Some things will grow, other things will shrink. The good thing is it seems like the gut bacteria are pretty good at self-adjusting. They'll fix themselves if you give them a little time. But my short answer is the mix. Although I will have someone who's gone to Mexico, I will say that if you get water that has some of the local E. coli, it can be pretty impressive. And that's just an infection. That's just, it's a great weight loss plan, but has some downside. Yes, ma'am.
There's such maladies like lactose intolerance,
Mm-hmm.
intolerance, which seem food-related,
Right.
and they're usually dealt with by restricting food or not taking. Could there be some way of dealing with them using bacteria?
Yeah, that's one thing that people are looking at is can we seed your gut flora with bacteria that can break down the sugar lactose. We have an enzyme to digest lactose, but most of the world's population turns it off around 4, 5, 6 years of age. Those of us of Northern European and a couple of African lineages keep it on because we were descendants of herders. So we, but yeah, who's doing that? There's a group, I can't remember who they are, working on can we feed you bacteria that will digest those milk sugars, break them into things that you can chew up for you, rather than have, that's what the lactose-free milk is. It's just milk that we've let some bacteria eat a little bit of. Perfectly safe, by the way. Yeah, well, the enzyme is bacterial. So, and there's two ways to do it. You can let them grow, or you can isolate the enzyme and throw it in. And the glucose, what is gluten? Thank you. That one's a little bit trickier. That seems to involve the immune system a lot more directly than the lactose intolerance. I would not be at all surprised if there isn't a microbial component, though, because we've been eating gluten for a long time, and it was fine, and suddenly it's not. It's like peanut allergies. Right? When I was a kid, we had one kid who had a peanut allergy. Now in my son's school, there's a peanut-free section, right? And that's not an act. I mean, there's something happening, and it's probably to do with what we have in our gut and how our immune system is getting educated. Something I didn't really talk about, your immune system learns things by dealing with the bacteria you encounter, and if it doesn't encounter the right things, it sees something like a protein from a peanut or ragweed pollen, right, that you breathe in your nose and thinks, you know, it's an Al-Qaeda invasion and goes crazy, and that's what most of the symptoms are. So, but yes, there is definitely work being done on that.
Can you comment on the benefits of having a very diverse community of bacteria in your gut?
Yeah, it's really important. An awful lot of chronic inflammatory conditions in the bowel are correlated with reduced diversity. Irritable bowel syndrome, ulcerative colitis, and it's weird because those seem very similar, but they're not. They happen in the same place, but the causes and especially the microbiology is very different in those two conditions. Diversity is really important.
How would you cultivate that?
We don't know. First of all, well, okay. One thing is not taking a lot of antibiotics when you don't need them. I keep saying this because I really, really want to make sure everyone gets this. If you have a mycoplasm in your lung, take the antibiotics. That's a much more pressing problem, and your microbiome will recover. If you take antibiotics all the time, that's different. One of the things that, it's a study that I didn't show you, 99-point-something% of the bacteria in your gut are these great big groupings. There's a tiny little sliver, really, really small percentages, but they seem important. People, the Chinese study, when they're looking at diabetes, it's not a big ratio that you could see on this graph. It's a really small change in a couple of really particular organisms that seem to throw everything out of balance. So I don't know how to cultivate that, and I don't think anyone else does, other than this. It sounds like, I feel like I'm like your mother, right? Eat a good and varied diet, get some sleep, stay hydrated, all the stuff that we, of course, know to do. But yeah, diversity in the gut is key. Lack of diversity in other places is key. So, yes, sir.
You had a graph up there about different bacteria in the guts of people from the Americas.
Yep.
And those were, what do you do with a country like the United States? How do you identify these people? And how many generations does it take to redo us?
That is a really important question that we don't, I keep saying we don't know the answer because we don't. How much of this is inherited, right? And one of the things, there's a paper that I read in preparation for this, talks about the influence of human genetics, right? Because it's not just our diet, it's our own personal genotype, right? We support different bugs. I mean, I don't want to give you the impression that the bacteria are all that matters in digestion. They don't, right? There's a vagus nerve that decides a lot of the stuff, and you have your own set of enzymes. So there is an interplay between those. We know that if you look at the biome of people in Japan, they have a bunch of bacteria that are good at digesting seaweed, because it's a big part of the diet. People in the Midwest don't. If someone from Illinois moves to Japan, in about a year and a half, we'll start seeing a few of those bacteria that digest seaweed, but it'll never get to the level of someone who was born there. What does that mean? And what about their kids, right? Some of the mouse studies, it seems like some of this is inherited. That if you look a couple of generations later, even if you've done everything the same, that some of the biome will be different. And so we don't, mice are a tricky model because their GI tract is very different from ours. So it's kind of hard to extrapolate some of those. But I think there's an immediate correction pretty quickly, sort of like the earlier question, in a matter of weeks or months, some of that shift happens. But I also think there are longer term things that we don't, there's no obvious reason why it shouldn't shift, but it doesn't. Some part of it stays how it was when you were a kid, even though you're not eating that way anymore. And that's in the long list of things that we don't clearly understand yet. Oh, one last question, and then I'll go over to small groups so the people can actually go on with their day.
Did I hear correctly that TB is on the rise?
TB is on the rise from where it was 10 or 15 years ago. It's still much lower than it was in 1900, obviously. Partly because TB, like most, it's a mycobacterium, not mycoplasma, sorry. Diseases of stress and crowding, and we have a bunch of profoundly antibiotic-resistant strains of tuberculosis, most of them that came out of Russia because of some bad management practices during the communist era. The other problem is that tuberculosis in the US is mostly a disease of people who are homeless and IV drug users. The antibiotic treatment you need to cure tuberculosis requires taking 4 pills a day for 6 months. The population that has tuberculosis is not in a place to organize their life to do that. So we get a lot of really incomplete treatments, and we've bred some really nasty antibiotic-resistant strains of tuberculosis. We also have a lot more people who are immunocompromised, both because of HIV and because of some of the treatments that we've been able to devise for organ rejection and chronic inflammatory conditions. And when your immune system is suppressed, which it has to be in those cases, you're more susceptible to tuberculosis. So yes, tuberculosis is up. It's not anywhere near where it used to be, but it's up from where it was in the early 90s. Okay, and I'll stop talking now, I promise. Thank you very much.